Hi everyone. Today we'll be discussing previous year prelims question paper of a very extremely important topic, which is science and tech, which belongs to GS paper 3, and we'll be covering all the questions from the year 2021. I'd like to mention something before we get into the questions and start today's analysis. See, we all read pretty much the same material, and in today's internet age, we all have equal access to it, right? However, you will notice that there are still gaps in our knowledge, and it is because the way we perceive things and the way our approach is. If suppose you get a question and you have the right approach to deal with it, it'll take you closer to the right answer, right? So the aim of this video is not just to tell you the right answers and shed away the wrong ones, but we will also be learning the that technique as to how to develop that right approach. All right. Okay then. Before starting, before start analyzing our questions, I would want all of you to look at this graph. This graph depicts the questions which were being asked in past 10 years in pertaining to science and tech domain. Let's try to infer the number of questions that are being asked in previous year question from this particular subject. In 2013-14, you can see there was similar number of questions which were being asked, and then in 2015 it got increased. Yes, it got decreased in 2016-17, but again 2018 में काफी questions पूछे गए हैं. So you can predict that there is this uncertain trend which we can analyze, right? So we must prepare ourselves for any kind of uncertainty UPSC throws at us, right? Therefore it becomes very important for us to to understand the concepts as well as the current part or the facts and everything, जिससे हमारे क्वेश्चन सॉल्व हो जाए. Okay, generally science and tech की अगर हम बात करें, so most of the question from this section is application based and mainly related to the current affairs. But mind you, अगर आपके concepts clear नहीं होंगे, you won't be able to have that command on the current affairs. Okay, so first you must clear your basics, you must clear your concepts, and then only you can have a proper command on current affairs. So when you prepare a particular topic in science and tech, you don't need to know everything about that particular technology you are reading, but you must know the basic features of it, right? And that's the procedure how we go through this paper. All right then, let's start with our first question. Our first question is, water can dissolve more substances than any other liquid because, all right, so the first thing we must do when we solve any paper is to read the question carefully, to understand the question carefully. Okay, so the question here is asking कि water can dissolve any substances more than any other liquid we have. Okay, and why is it so? Is it because it is dipolar in nature, or is it because it is the good conductor of heat, or is it because it has the high value of specific heat, or it is an oxide of hydrogen? Okay, for that I want you to look at this figure. This figure I've put for your more clarity. See, you can see that water molecule have a polar arrangement of the oxygen, right? Of the oxygen and the hydrogen atom, right? One side hydrogen यहाँ पे present है, which are positively charged, as you can see here, and the oxygen is present on the other side, which is negatively charged. And this entire arrangement allows the water molecule to become attracted to many other different types of molecules, and also it makes it polar in nature. Hence, which one should be the correct answer? The correct answer should be option number A, which is it is dipolar in nature, and that is why it can dissolve more substances than any other liquid we have. And for these type of questions, you must read your NCERTs properly. At least once you must read NCERTs. एक दो बार आपको पढ़ना चाहिए all the basic NCERTs of all the subjects. Then only you can have the command on these small basic things. All right, moving ahead with the next question. Question number 2 says, with reference to street lighting, how do sodium lamps differ from LED lamps? All right, before getting to the options of it, we must understand the basic difference between sodium lamps and LED lamps. So you all must have heard of HPS lamps, which are called high pressure sodium lights, जिनको हम sodium lamps भी बोलते हैं, right? They were the most efficient technology, but वो पुरानी technology हो गई है. So these sodium lamps are the technology of 1970, ठीक है? While LED is a modern day technology, we all must be knowing this as we all use these appliances which have LED in it in our home, right? So we everyday use this technology, okay, the LED technology. Although sodium vapor lightning, this sodium lamp wala technology, it beats virtually every other technology in terms of energy efficiency, but it loses out to LED. It does not have better efficiency than LED because and LED, mind you, LED is not just energy efficient, but LED also has a longer lifespan than the sodium lights, okay. And apart from this, LED also provides an incredibly better variety of high color options. जबकि जो sodium lamps थे, if you have noticed, sodium lamps वाले जो bulb हम घरों में use करते हैं, they kind of eliminate a monochromatic black appearance, right? So whenever we use these sodium lamps, they eliminate they eliminate this, they have this black color appearance around them, while LED lamps में ऐसा कुछ नहीं होता है. Now let's read the options. Option number 1 says sodium lamp produce light in 360 degrees, but it is not so in case of LEDs. This is an correct statement. ठीक है, सोडियम लैम्प्स 360 डिग्रीज में लाइट देता है, बट इट इज नॉट सो इन केस ऑफ एलईडी लैम्प्स. व्हाई सो? बिकॉज एलईडी टेंड्स टू डायरेक्ट द लाइट डाउनवर्ड्स rather than the 360 degrees. All right. Second option says street lights में sodium lamps have a larger lifespan than the LED lamps. So this is an incorrect statement because I've just told you that LED lamps, they are not just energy efficient, but they also have the longer lifespan than the sodium lamps or the older lamps we used to use. The third statement says spectrum of visible light from sodium lamps is almost monochromatic, while LED lamps provides a significant color advantage in the street lighting. Yes, this is a very correct statement कि black appearance या फिर एक monochromatic appearance होता था sodium lamps के आसपास, which LED lamps does not have. LED lamps provide a range of or a variety of high color options, right? So which one should be the right answer? Because 2 is the wrong answer, therefore C should be the right answer of this question. I hope it is clear to you. Moving ahead with the next question, question number 3. Which of the following is the reason why astronomical distance are measured in light years? Astronomical distances ko we measure in light years. What could be the reason behind it? The first reason is distance among stellar bodies do not change. No, they do. So this cannot be the right answer. Gravity of stellar bodies does not change. Gravity also changes. Light travels in a straight line. No, never. It is an incorrect statement. The last statement says speed of light is always the same. So we know that when we measure measuring in light years also allows the astronomers to determine how far back in time they are viewing, because light takes time to travel to our eyes. Everything we view in the night sky has already happened. So and because the speed of light is empty, the speed of light in empty space या फिर vacuum, it never ever changes, and this is precisely the reason why astronomical distances are measured in the light years. I would want you to look at this diagram and just pause the video right here and look at the seconds it takes from Earth to moon to sun and all these bodies. Okay, just pause the video and look, just have a glance at this picture. Just look at the picture. You don't need to memorize everything. Just look at it once. Just have a glimpse of it once. What should be the right answer for this? That the speed of light does not the speed of light in empty or the vacuum spaces does not change or never changes. Therefore, D is the right answer for this particular question. Moving ahead with the next question, question number 4. In pressure cooker, the temperature at which the food is cooked depends mainly upon which of the following? Okay, whether it is dependent upon the area of the hole in the lid, or it is also dependent on the temperature of the flame, or is it also dependent on the weight of the lid? So it is dependent, the temperature at which the food is cooked, it is dependent majorly on all the 3 mentioned here. How? I'll tell you. See, the area of the hole in the lid, what it controls how much steam is let out, and thus pressure inside the cooker. Therefore, it indirectly affects the temperature. Therefore, first option is the correct option, that the area of the hole in the lid is one thing which, which basically the temperature, it depends on this, it depends on the area of the hole in the lid. Second, it says whether the temperature of the flame, ये एक dependent variable है या नहीं, the temperature. Let's look at this also. So this also plays an important role, hence making this option also correct option. Third, it talks about the weight of the lid. So yes, definitely the weight of the lid also helps to maintain the steam pressure, thus influencing the temperature directly. So all of these, all the three options impacts the temperature directly or indirectly, hence making all the statements correct in this question. Moving ahead with our next question, question number 5. Bisphenol A, a cause of concern, is a structural or a key component in the manufacture of which of the kind of plastics? Okay, let's see. So before we get into the options, what is it related to, we must understand what bisphenol A is all about. Bisphenol A है क्या? Let us look at some of the important or the major properties of it. So bisphenol A it is a very important compound, and it is basically used in the manufacturing of various plastics, mind you, in what, in the plastics, and it is poorly soluble in water. और इसके काफ़ी सारे हेल्थ कंसर्न्स भी हैं, एज इट इम्पैक्ट्स द ब्रेन, ठीक है, प्रोस्टेट ग्लैंड्स को भी ये काफ़ी इम्पैक्ट करता है, स्पेशली ऑफ द फीटस इन द इन्फेंट्स. सो बेसिकली ये चिल्ड्रन को ज़्यादा इम्पैक्ट करता है, द ब्रेन ऑफ द चिल्ड्रन और द प्रोस्टेट ग्लैंड ऑफ द children. But the question is asking that what is it related to, or you can say it is a key component in the manufacture of which of the following plastics. All these are the examples of plastic, but we have to identify कि BPA इसमें से किसमें use होता है. So it is used in polycarbonate, hence making B the right option. What are polycarbonate plastics? So they are also used in making plastics only, but what kind of plastics? They are used in making hard plastics, hard plastic items basically. Like for example, bottles हम बनाते हैं polycarbonate से, food containers हम बनाते हैं, tableware बनाते हैं, right? Okay, so there's one approach to deal with such kind of questions, that if you have heard of bisphenol A, if you have read your chemistry books, if you have read your NCRTs, and if you know the answer of it, just tick the question. If you do not know the answer of it, I do not think you can, you know, if you're not a chemistry expert, you can anyhow reach to these answers. You can anyhow connect something or logically reach to the answer, or you can make any educated guess. So if you know it, just mark it. If you don't know it, move ahead with the next question. All right. Okay, then B is the right answer for this. Moving ahead with our next question. Next question talks about triclosan. Triclosan considered harmful, mind you, it is harmful when exposed to high level for the long time, and is most likely present in which of the following? Triclosan is present in which of the following? Is it present in the food preservatives or food ripening substances or reused plastic containers or toiletries? Again, अगर आपने कभी भी न्यूज़पेपर में या anywhere, if you have heard of such term, you can definitely give it some time, give this question some time. If you have never heard of such term, if UPSC asks such terms और आपने कभी नहीं सुना हो, you have no idea about what triclosan is, you must leave the question. No need to, you know, कि कुछ भी आप तुक्का मार के you're just marking any of the options. There are high chances कि वो गलत ही होगा, so you must leave such questions. बाकी ट्राइक्लोसान जनरली इज रिलेटेड विद द टॉयलेटरीज, सोप्स. तो टॉयलेट्स में बहुत सारी जो भी चीज़ें यूज़ होती हैं, triclosan is related to them. So it is used in the hand washes, soaps, right, and all these. And it was published in the Indian Express का एक आर्टिकल में ये पब्लिश हुआ था, 2019 की ये बात है. एंड आफ्टर one and a half year, UPSC has asked this question. So make sure you read such terms, or you read your newspapers carefully at least whenever you come across such new or any term which you are not aware of. Make sure you note down them. उनके बारे में आप एक दो लाइन लिख लीजिए, उनको समझ लीजिए. You never know कि पेपर में वो कब आपके काम आ जाए. Right? Moving ahead with the next question, question number 7. It says, with reference to recent developments regarding recombinant vector vaccines, consider the following statements. Okay, recombinant vector vaccines. This is an important topic and an important question, and we will be dealing it in detail. Don't worry, आपको सब clear हो जाएगा. What are recombinant vector vaccines, and of course this question also. So सबसे पहले to deal with certain such kind of questions, our basics must be clear for these type of questions, right? As I have told you in the beginning of my lecture only, that to have a command on current affairs, you must be clear with your static portion, with your basics. Then only you can improve your current affairs, warna current affairs nahi honge. So do not ever jump to do newspapers or current affair magazines first. First prepare your static portion, ठीक है, उसके बाद ही start reading newspaper or whatever current sources you are following. All right. To understand this question, we must understand what is recombinant vector vaccine, and for that I'd like to break this term into two words. The first would be the recombinant itself. So recombinant का मतलब क्या होता है? Recombinant in general means anything which is formed by combining two things, and that is why we call it recombinant. All right. Now what is vector? Vector क्या होता है? Vector is anything, it's basically it's like a vehicle which is used to carry a foreign genetic material into the host cell. Okay, so इन दोनों चीज़ों को combine करके the recombinant and the vector, we make vaccines using this technology. And the famous examples of you can say recombinant vaccines are the hepatitis B vaccine and the polio virus. I repeat that recombinant vector vaccines, they are basically used by combining two things, or we can say that they are made using viruses or the bacterial or the yeast cells to manufacture the vaccines. Now you must understand, or you must ask, that what is the exact technique behind making the recombinant vector vaccines, or how do we make such vaccines? Okay, for that let me take you to this a very beautiful diagram जिससे आपको बहुत this is a very clear diagram which will clear most of your doubts. Okay, so what is happening here, see it properly. Let me tell you what exactly we do in this particular technique. So what exactly we are doing is we're taking a small piece of DNA from the virus or from the bacteria, which we are calling the donor DNA, right? Okay, so we are taking this DNA from the virus or from the bacteria against which we want to protect, and we ligate it. Ligate means we join it with the another piece of DNA which we have taken from the vector, and this vector piece of DNA is not harmful to us. So basically what we are doing, we are taking one piece of DNA which is harmful to us, and we are taking another piece of DNA from the vector which is not harmful for us, and we are ligating both of them, as in we are joining both of them, and then we will put them to replicate, और इसके unharmful copies बन के हमारे पास, not harmful copies हमारे पास जो भी बन के आ जाएंगी, and then we can inject it, right? We can inject it in human body, and the moment we will inject it, after some time it will produce antibodies against the particular virus or a particular bacteria, hence providing us the immunity. So this is the basic role of it. For those who are still not clear with it, let me tell you, or let me repeat this in a very layman's language. So what we are doing here, on one hand, we are taking a piece of DNA from the infected cell, right? And on the other hand, we are taking a piece of DNA from the non-harmful, or the vector, or the which is not harmful for us. We are joining both of them, we are ligating both of them, we are putting them into the bacterial cell, we are letting them to replicate, और इसके बहुत सारे ऐसे जो safe, safe copies हमारे लिए बन जाएंगी. We will inject these safe copies into the human body, and it will produce antibodies, and in turn give us the immunity against any particular virus or any particular bacterial diseases. All right, I hope it is clear to you. I hope the entire technology is clear to you. Now let us come to the options. Option number 1 says the genetic engineering is applied in the development of these vaccines. Yes, definitely, the entire process I've just told you, this entire process is related with, or is a part of, genetic engineering only. So option number 1 is the correct answer. Is the correct option. Option number B says bacteria and viruses are used as a vector. So yes, bacteria and viruses also we use in vectors. हाँ, वो अलग बात है. We turn them in such a form that it becomes safe for the introduction into the human body. So both the answers are correct. Both the options are correctly mentioned here. Therefore, C is the right answer for this question. Moving ahead with the next question, question number 8. In the context of hereditary disease, consider the following statements. Okay. Like my approach is, before moving on to the options, I always make sure that you understand the question first. So keeping in mind the same approach, let me first explain or make you understand what hereditary diseases are. Hereditary disease का क्या मतलब होता है, and then we will read the questions. Okay, so hereditary disease basically जब कोई भी genetic disorder हमें हो जाता है and we inherit that genetic disorder either from one of our parent or both of the parents is called hereditary disease when we are acquiring it from our parents. Right? Like for example, a very famous example of hereditary disease is sickle cell anemia. Sickle cell anemia क्या होता है? What happens in sickle cell anemia that the shape of our RBCs they get deformed और वो sickle जैसे shape की हो जाती हैं. And then what happens अगर वो sickle जैसे shape के हो जाते हैं, that shape basically blocks the blood and oxygen flow to our organs, and due to the lack of oxygen rich blood, it can damage our nerves, it can damage our kidneys, liver और बहुत सारे parts को वो damage कर सकता है, and it can even be fatal. So hereditary diseases can even be fatal to us, right? Now let us read out the first statement. The first statement says, passing on the mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. Okay, I'm sure ये बहुत ही कॉम्प्लिकेटेड सा टॉपिक दिख रहा, ये बहुत ही कॉम्प्लिकेटेड सा ऑप्शन आपको दिख रहा होगा, एज इट इज टॉकिंग अबाउट सो मेनी डिफरेंट न्यू थिंग्स. इट इज टॉकिंग अबाउट माइटोकॉन्ड्रियल डिजीजेस, इट इज टॉकिंग अबाउट MRT, which is mitochondrial replacement therapy, it is talking about in vitro fertilization, and you all must be having no idea about what this topic is talking about unless you have read biotechnology in your graduation. Okay, so don't worry, I'll make it simpler for you, I'll make it easier for you, I'll make you understand mitochondrial diseases क्या होते हैं, MRT क्या होती है, इस थेरेपी के क्या बेनिफिट्स हैं, और इन विट्रो और इन विवो फर्टिलाइजेशन क्या होता है. So first thing first we should understand what mitochondrial replacement therapy is. So in simple word, what is happening, as the name indicates, mitochondrial replacement. So mitochondria is getting replaced here. Mitochondria, which is the powerhouse of our cell, is getting replaced here with something. That is why we are calling it mitochondrial replacement, right? So it is the replacement of mitochondria in the cells to prevent a particular disease, right? And it is a form of in vitro fertilization. Now, what is in vitro fertilization? See, there are two kinds of fertilization. One is in vitro. In vitro means when we fertilize the eggs and the sperm in the lab, in the laboratory, we do the fertilization. On the other hand, there is another kind of fertilization which is called in vivo fertilization. What happens in vivo fertilization? When the research work or this entire fertilization is done within a living organism is called in vivo fertilization. Okay, but we must stick to our mitochondrial replacement therapy. I am sure a picture would help you better understand the MRT or the entire technology. See what is happening here. Just focus here on this diagram. So what's happening here? So basically, where the future baby's mitochondrial DNA comes from the third party, इसी को हम MRT कहते हैं. But let me go into the detail of it and let me explain you this entire diagram. What is it depicting here? So this is the mother and this is the donor mother. What is happening here? First, we transfer the mother's nuclear genetic material from an egg with diseased mitochondria into a donor's egg which has a healthy mitochondria. So if suppose she's a mother, she has some disease, all right, we will take out this sick mitochondria, okay, we will remove this sick mitochondria. On the other hand, she's a donor mother, okay, or she's a donor, not mother, she's a donor person, she's a donor, right? इनका भी हम healthy mitochondria लेंगे and we will introduce this sick mitochondria into the healthy mitochondria, right? So this is basically what we are doing, and this healthy mitochondria in the donor egg can allow the normal development, right? And it will stop the transmission of that particular mitochondrial disease जो कि mother को है, hence preventing it from transmitting into the child, right? And then आगे fertilization वगैरह होता है, but we must focus here only that what is happening in MRT or the mitochondrial therapy. Let me make it more lucid for you. So there's a mother, there's a donor. मदर के माइटोकॉन्ड्रिया में कुछ प्रॉब्लम है, ठीक है, उनके मदर्स न्यूक्लियस को हम निकालेंगे. वी विल इंट्रोड्यूस दैट मदर्स न्यूक्लियस इनटू अ हेल्दी माइटोकॉन्ड्रिया, और यहाँ पे दिस entire healthy mitochondria will give the ambience, will give the environment to the mother's nucleus in such a way that the healthy things will produce, and this healthy mitochondria in the donor egg can allow for the normal development, and it will stop the transmission of the mitochondrial diseases. Hence we can prevent any kind of mitochondrial diseases, right? Okay, so yeah, the next question, the next option says a child inherit mitochondrial diseases entirely from the mother and not from the father. Okay, so there's a very important point I'd like to mention here, that while the nuclear DNA comes from both the eggs and the sperm, but mitochondrial DNA जो होता है, the DNA which is present in the mitochondria is largely passed on from the mother's egg only, ठीक है? So father does not have any kind of role in it. अगर mitochondrial diseases किसी बच्चे में हो रहा है, it is because कि mother में भी वो particular diseases थे, so it passes through the mother only. So this point you have to remember. Hence this is a correct statement, but first statement here is incorrect. Why? Because what happens here, that this entire step or this entire procedure has to be done before the fertilization. The question is asking it can done either before or after the in vitro fertilization of egg. नहीं, in vitro fertilization से पहले हमें ये पूरा प्रोसेस करना पड़ेगा, so as to prevent the any kind of mitochondrial diseases, hence making the first statement incorrect here. That is why we are left with option number B, which is correct answer of question number 8. Moving ahead with question number 9. Question number 9 says, consider the following: bacteria, fungi and virus. Which of the following or which of the above can be cultured in artificial medium or the synthetic medium? I am not sure who all of you knows the meaning of the artificial medium or the synthetic medium, but let me tell you that there are two kind of cultural media. First is the natural media or the synthetic media. What happens in natural media, that it includes all the natural or the biological fluids, as for example plasma hota hai, usme serum hota hai. Whereas synthetic medium में हम organic, inorganics, हर तरीके के nutrients या फिर media use करते हैं. The question is asking which of the above can be cultured in artificial or the synthetic medium. हम इन तीनों में से क्या synthetic medium में culture कर सकते हैं? But before that, let me show you this diagram which says that we are using a culture, we are using a media which is molten agar. इसमें हम bacteria का solution डाल रहे हैं. We are incubating it for whatever time it takes actually different time for different bacteria, and then the colonies will be formed of the bacteria, and we can we can further use it. But this is the basic process behind how we culture things in the labs. Okay, so the question was asking कि हम इन तीनों में से what all could be cultured. So let me tell you, the bacteria could be cultured, we grow this in lab. The fungi could be cultured, we definitely grow this in ये सब biotech labs में grow हम करते हैं. But the viruses cannot be, because viruses are grown in living cells, right? They need host cells. हमें पता है viruses को grow करने के लिए host cells या living cells की ज़रूरत होती है. We cannot artificially grow them. That is why third option is the incorrect option. हम इस question को इस तरीके से भी solve कर सकते थे अगर आपको वायरस के बारे में ही पता हो. If you know just about the virus, that virus could not be grown into such environment or such condition or in the synthetic media. So you could have eliminated option number B, C and D. Hence you would have only left with option number A, which is bacteria and fungi only could be grown in the artificial or the synthetic medium, right? Moving ahead with the next question, question number 10. The term ACE2 is talked about in the context of what? ACE2 किसके बारे में हमने सुना है in the recent past. So is it a gene which is introduced in the genetically modified plants, or it is a development of India's own satellite navigation system? No, it is nowhere related to the navigation satellite system. If you read your newspaper carefully, and let me tell you that anything which is being repeatedly given in the newspaper, or they're mentioning it almost every week, वो चीज़ें तो at least आपको पता होना चाहिए. Radio, is this used for wild tracking? No. Is it a spread of viral disease? Yes. So you all must have heard this term ACE2, which is the full name of it would be ACE2 receptor. Corona के time में आपने बहुत बार सुना होगा ACE2 receptors. So it is related with the spread of viral disease. Here you can see these are the ACE2 receptors, and they basically what they do, these are the enzymes that generate small proteins that are useful for the cells, और इन्हीं के through the virus enters the, this is the virus, this is the body, and this is that receptor, ACE2 receptor, और इन्हीं के through the virus enters the human body, and that is why it was in news so much while the corona thing was there. ये बहुत ज़्यादा news में था, ACE2 receptors, right? Okay then, moving ahead with the next question, question number 11. Consider the following statements. Adenovirus have single-stranded DNA genomes, whereas retrovirus have double-stranded DNA genomes. Okay. Common cold is sometimes caused by adenovirus, whereas AIDS is caused by retrovirus. All right. Again, like I always say, that you must first understand the meaning of whatever line is given, whatever statement is given in your question. Here there are 2-3 things we must understand. First, we must understand the difference between adenovirus and the retrovirus, and then we must understand what are single stranded and what are double stranded DNAs. ठीक है? Adenovirus और retrovirus के बीच में difference समझने के लिए we must know a very important word, which is envelope. Let me show you this through this diagram only. Suppose this is the structure of the virus. This purple color, you can see these here only, the outer covering, basically we call it envelope, right? And that is the distinction between the adenovirus and the retrovirus. I'll explain you how. Okay? So what happens in adenovirus, the main difference between them, basically, as I've told you, it depends on whether or not the envelope is present outside the virus. Now you may ask कि ये envelope क्या है? So this is basically this is just the outer protective layer of the virus. But what is the significance of it? इसका काम क्या होता है? What is the importance of it? It is of huge importance, because this protective layer only makes the virus more resilient, and it increases the risk of infection in the human body. ठीक है? That is why it holds a huge importance. Now we must also ask कि अभी एडिनोवायरस में ये प्रेजेंट होता है या रेट्रोवायरस में दिस एनवेलप इज प्रेजेंट. सो एडिनोवायरस डज नॉट हैव दिस एनवेलप. ठीक है, एडिनोवायरस में ये एनवेलप नहीं होता है, वेयर एज रेट्रोवायरस हैज दिस एनवेलप. ठीक है, अगेन आई मीन नेक्स्ट थिंग which is to be understood here is the difference between single stranded and double stranded DNAs. Okay, DNA हमें पता है, it exists in 2 forms. First is the single stranded and second is the double stranded DNA. Let me take you to that, to one of our diagrams. Yeah, to this diagram. यहाँ पर आपको और भी अच्छे से clear हो जाएगा about this strands of DNA. DNA हमें पता है, DNA के पास nitrogenous bases होते हैं which form hydrogen bonds with each other. Nitrogenous bases are these bases, the A and T. A stands for adenine, T stands for thymine. So these are the nitrogenous bases, and due to the hydrogen bonding between these base pairs only of the 2 strands, the structure of the double stranded DNA is more stable than the single stranded DNA. ठीक है, and most of the organisms they use double stranded DNA only to encode the genetic information, क्योंकि double stranded DNA जो है ज़्यादा stable होता है. but ये I have shown you this picture for you to understand that these are the nitrogenous bases and these are linked with these are paired up with the hydrogen bond, making the entire structure of DNA more stable. Right? Let's go to our question again. Okay, so the question says that adenovirus have single-stranded DNA, whereas retrovirus have double-stranded DNA. Let me tell you that yes, adenovirus is a group of DNA virus, whereas retrovirus, you have to understand this and you must memorize this, that retrovirus belongs to the RNA virus. ये DNA viruses DNA genome से belong ही नहीं करता है, making the first statement highly incorrect here. ठीक है, हम डबल स्टैंडर्ड या सिंगल स्टैंडर्ड तो तब देखेंगे व्हेन इट बिलोंग्स टू डीएनए ओनली. इट डज नॉट बिलोंग टू डीएनए ओनली, इट बिलोंग्स टू द आरएनए फैमिली और द आरएनए virus, right? So first statement is the incorrect statement here. Question number 2 say option number 2 says common cold is sometimes caused by adenovirus, whereas AIDS is caused by retrovirus. I'll tell you what could be the trick you could be using in this particular option. So if you ever heard of even little bit about HIV AIDS, आपको पता होगा that it is caused by single stranded RNA. It is caused by single stranded RNA, and a while ago we have just read that retrovirus is also an RNA virus, and HIV AIDS is also a RNA virus. So you can link the retrovirus with the HIV as both have RNA as their genetic material, and you can reach your answer. Okay, so this is also one of the approach to reach your answer even if you are not aware of the entire statement given in your question. Okay, so first is incorrect, second is correct, therefore B is the right answer for question number 11. All right, moving ahead with the next question. Question number 12. Which of the following is used in preparing a natural mosquito repellent? Mosquito repellent में कौन सी ग्रास हम इनमें से यूज़ करते हैं? कांग्रेस ग्रास? नहीं जी, हम कांग्रेस ग्रास नहीं यूज़ करते. कांग्रेस ग्रास को हम कैरेट ग्रास भी बोलते हैं, and it is native to, if you know, it is native to American tropics, right? And it causes skin and respiratory problems. Skin allergy cause कर देती है. You all, अगर आपने कभी इसको touch किया होगा, skin allergy cause कर देती है. It also causes a lot of respiratory problem. So it is not a mosquito repellent, rather it is another kind of problem only. Elephant grass? No, elephant grass is also not a mosquito repellent. Elephant grass generally हम इसको Uganda grass भी बोलते हैं. It is because it is native to African grasslands, and it is used in manufacturing of paper or the handicrafts, etc. It is not a mosquito repellent. The third is the lemongrass, and fourth is the nut grass. So nut grass is also not used as a mosquito repellent. Okay? It is basically used in this various kind of skin treatments, but it is not a mosquito repellent. And therefore, we are only left with option number C, that is lemongrass. To show you the picture of lemongrass, see, this is the lemongrass, right? और इसको हम as a mosquito repellent use कर सकते हैं. Right? Moving ahead with the next question. Question number 13. Bolgard 1 and 2 technologies are mentioned in the context of what? Okay. First, let us understand कि ये technologies क्या हैं. फिर उसके बाद खुद ही समझ आ जाएगा what are they related with. We'll come to the answer automatically. So it was in news some time ago. Recently what happened, our center considered writing of trait value which were charged from the farmers for Bolgard 2. It is basically a variety of BT cotton. And why center did this? To ensure the seeds are available to farmers at an affordable prices. See, though there are many benefits of this, like Bolgard 2, जो cottons होती हैं, they provide protection for cotton against the destructive bollworms, in a lot of other kind of infestations also. Okay? But at the same time, it has certain disadvantages also. Like it is expensive. So it is a kind of BT cotton only. Okay? BT cotton, which is definitely related with what? Which is a genetically modified crop plant. So इससे हम answer पे तो directly we have reached that this is a developing genetically modified crop plants. But what is more important for our examination perspective is to understand about genetically modified crops. We will not be able to read the details of GM crop, but because that's a very big topic. It's an important topic for some another day. But today, let me just touch upon the topic. So what are GM crops? So genetically, जिनके gene को हमने artificially insert कर दिया है, from a so basically, let me just simply read out for you. A crop which has a gene artificially inserted into it from some another species, even unrelated, to give it some desired properties. In short, in easy language, genetically modified crops are basically जिनका जो DNA है, the DNA of which has been modified using the genetic engineering methods. We call them genetically modified crops. If we talk about the status, or when did India get its first GM crop? So it was first of all, it was BT coated, ball guard 1 जिसको हमने बोला है, which provides immunity against a lot of infestation including the pink bollworms, or initially it was developed by Monsanto. So I hope it is clear that it is related with the genetically modified technique or a genetically modified plant. Right, moving ahead with the next question, question number 14. Which of the following statements are correct regarding the general difference between a plant cell and an animal cell? Again, a very basic question direct from our NCERTs. That is why I am again and again telling you that you must first clear your basics, first clear your concepts. तभी आपके, do not worry about the current affairs, वो हो जाएंगे. First you must clear your basics, clear your concepts, clear your NCERTs first, and or do not worry about the current affairs. Okay, they will be followed. So they're asking about the general difference or the major difference between animal cell and the plant cell. Okay, the first option says plant cell have cellulose cell walls while animal cells do not have. Yes, this is a correct statement. Let me take you to this diagram. So yeah, the option also says that plant cell has cell wall. Hanji, plant cell has cell wall. Animal cell, no, it does not have any cell wall. You can take a picture of it or you can take a screenshot of it. This, I'm telling you, this is going to help you so much. This is such a clear diagram जो आपके दोनों distinctions as well as similarities दोनों को ये depict कर रहा है. Very, very important diagram. You must take a screenshot of it and you must keep it with you. You must revise it just before your exam. So plant cell have cellulose cell wall while animal cell do not have. We have just seen it is a correct statement. Cell wall का basically काम क्या होता है? It provides a structural support to the entire cell. The second statement says plant cell do not have plasma membrane while unlike animal cell which do have. So this is an incorrect statement. It says plant cell do not have plasma membrane. Plant cells actually have plasma membrane. ठीक है, plant cells के पास plasma membrane होती है. And what is the function of that membrane? What is the function of that plasma membrane? It basically provides the tensile strength and protection against the mechanical or the osmotic stress to the cell. So it provides you that protection. ठीक है. The third statement says mature plant cell has one large vacuole while animal cell में बहुत छोटे छोटे vacuoles होते हैं. This is a correct statement. It is mentioned over here as well that plant cell has one large vacuole while animal cell either they will have no vacuole or they will have many small vacuoles. Now you must ask कि इन vacuoles का काम क्या होता है? So these vacuoles basically they helps to sequester the waste products. ठीक है, जो भी वेस्ट प्रोडक्ट्स प्रोड्यूस होता है एक सेल में, दे हेल्प्स टू सीक्वेस्टर दैट वेस्ट प्रोडक्ट एंड मेंटेन द वाटर बैलेंस इन द सेल. दिस इज द मेजर फंक्शन ऑफ व्हाट? ऑफ द वैक्यूल. सो वी आर लेफ्ट with option number 1 and 3 which is correct, therefore making C the right answer for this question. Now talking little more about this particular diagram, hence this is very important. Let me tell you the characteristics which are common in both the cells, the plant cell and the animal cell. The first is the mitochondria. We know it is present in both the cells and it is the powerhouse of the cell. The entire cell gets the energy from the mitochondria itself. The second is the Golgi apparatus which is present in both the cells against Golgi apparatus. The third is the rough and the smooth endoplasmic reticulum, the rough and smooth ER also we call it. And what is the function of ER? Smooth ER, जो smooth endoplasmic reticulum होता है, they are basically or they are largely associated with the fat manufacturing, whereas the rough ER or the rough endoplasmic reticulum, they are largely associated with the production of proteins. And this difference also could be asked in your examination, so you make sure that you write it down, the difference between smooth and rough ER. Next is nucleus. We know the entire genetic material is inside the nucleus only, right? And cytoplasm basically it is the medium for all the chemical reaction. All the chemical reactions which are happening in the cell are taking place in this liquid only or taking place in this cytoplasm only. And lastly, ribosomes, which is present in both the cells, plant cell and the animal cell. But what is the major function of it? Ribosomes basically they're made up of RNA and protein and they serve as a site of protein synthesis only, right? All right, then moving ahead to our next question, question number 15. In the context of recent advances in human reproductive technology, pro-nuclear transfer क्या है या फिर pro-nuclear transfer किस में use होता है? Okay, so to read this topic, we need to go to our previous slides wherein we talked about MRT technology or the mitochondrial replacement therapy technology. वहाँ पर चीज़ें जो हमने पढ़ी थी, so pro-nuclear transfer is associated with MRT therapy only, right? We've read about that. I'll take you to that previous slide and that diagram only. But before that, let me tell you that what happens in the mitochondrial therapy or the mitochondrial replacement therapy, or we also call it mitochondrial donation, because we are a donor female is donating a safe or the fresh or the healthy mitochondria to the mother, ठीक है, जिनको mitochondria में कुछ defects हैं. So there are two technologies through which we can achieve the MRT technology, right? First of all, let me take you to that diagram only so that you'll be able to connect in a better way. Okay, so this was the diagram, this was the MRT technology. So there are basically two technologies through which we can achieve this entire thing. The first is the maternal spindle transfer and second is the pro-nuclear transfer. Maternal spindle fiber or pro-nuclear transfer, you don't have to go into the details of this technologies, but आपको थोड़ा सा पता होना चाहिए just to you know have this clarity in your mind. So both are similar only, both are used in MRT only, both are similar only, but there's one slight difference that what happens in pro-nuclear transfer. Pro-nuclear transfer में हम क्या करते हैं? It basically involves fertilizing the mother's egg first and then transferring the nuclear DNA to the fertilized donor egg. Okay, which is containing the healthy mitochondria, right? From which the original nuclear DNA has been removed, right? I repeat that what we are doing in nuclear technology, this pro-nuclear transfer, that what we do, that the entire thing involves fertilizing the mother's egg first. We fertilize the egg first. जब हम pro-nuclear तरीके से transfer करते हैं चीज़ों को, we fertilize the egg first and then we transfer the nuclear DNA to this donor or the fertilized or the donor egg containing the healthy mitochondria from which the original nuclear DNA जिनका remove कर दिया गया है. There's also another diagram attached with that question only. Let me show you that. Yeah, this was the question we were doing, pro-nuclear transfer, and this is the diagram related to it. Same thing happening in it कि हमने चीज़ों को पहले fertilize कर लिया है. We have made this embryo out of it, right? And then we have taken out the nucleus, which is, yeah, we have taken out the nucleus, ठीक है. These are the unhealthy mitochondria, ठीक है, जो कि parents का था, unhealthy mitochondria, and here we have the donor's nucleus. We will fuse both of them and hence, और फिर आगे क्या होगा? It will provide that ambience or that environment to the unhealthy to the unhealthy part, that it can grow healthier, right? Hence preventing any kind of mitochondrial diseases happening to the child, right? Whereas maternal में जो हमने दो तरीके पढ़े थे, first one was the maternal spindle transfer and this was the pro-nuclear transfer. Maternal में क्या होता है? We use the unfertilized egg. So this is the major difference, that pro-nuclear transferring करते time we use the fertilized egg and do the same process, but in case of maternal spindle fiber we use the unfertilized egg, and that's the major difference. बाकी दोनों ही जो हैं, both of them are related with the mitochondrial replacement therapy only, right? Okay, so these kind of technologies are very important. As you can see, two questions were asked from the same topic. Mitochondrial से ही दो question आपसे पूछ लिए गए हैं, but एक ही in the same year, so you can understand the importance of it. Moving ahead with the next question. Next question is, what is the importance of using pneumococcal conjugate vaccine? Again, we first should understand what are pneumococcal conjugate vaccine, and like I always do, I will break the word into two parts. First would be the pneumococcal, second would be the conjugate. But before that, I want to tell you कि ये question पूछा कहाँ से गया है and why it was asked. So recently, what had happened that our Union Health Minister launched the expansion of this thing, pneumococcal conjugate vaccine, under a very important program. Even वहाँ से भी question could be asked from in your examination. That program is called Universal Immunization Program, UIP we call it, and it is a part of Azadi Ka Amrit Mahotsav, and this entire program of PCV, which is pneumococcal conjugate vaccine, is going to reduce the child mortality due to pneumonia by around 60%. It is predicted that it will be reducing the child mortality by 60%. और अगर हम अपने इंडिया के कुछ फिगर्स की बात करें, so in our country around 60% of the deaths in children, they occur due to pneumonia only. So you can imagine how big of a plight this is. So PCV, which is pneumococcal conjugate vaccine, will greatly help. This will greatly help overcoming this particular disease. Now understand what are pneumococcal conjugate vaccine. Pneumococcal basically, pneumococcal disease. It refers to any illness which is caused by the pneumococcal bacteria. But there's one thing you must remember here कि ज़रूरी नहीं है this pneumococcal is caused by bacteria only. It can also be caused by viruses and fungi. ठीक है? But majorly it is caused by bacteria. That is why we call it pneumococcal bacteria. It is a bacterial disease. ठीक है? On the other hand, what do we mean by conjugate? This word conjugate or the conjugate vaccine simply means that vaccines which are made using the combination of two different components. So they're basically made using the pieces from coats of bacteria, which are chemically linked to what? Which are chemically linked to the protein, and the combination is used to make the vaccine. I hope the meaning of this word pneumococcal conjugate vaccine is clear to you now. Now let us read the options. Option number 1 says, these vaccines are effective against pneumonia as well as meningitis and sepsis. Exactly, this is correct. Meningitis is related with the brain problems. The second thing is dependence on antibiotics that are not effective against drug-resistant bacteria can be reduced. So yes, this can be reduced. This dependence on antibiotic could be reduced. The third says these vaccines have no side effects. एक तो ना ये जो बहुत ही आपको extreme चीज़ें दी जाती हैं ना एग्जाम में कि they can't have any side effects only, or these vaccines do not have any side effects only. Make sure that you take a pause and you think over it. ठीक है? ऐसा कुछ ही होता है जिसके साइड इफेक्ट्स नहीं होते हैं. सो दे डेफिनेटली दे हैव अ लॉट ऑफ साइड इफेक्ट्स. दे हैव फीवर्स हो जाता है, जैसे फीवर होता है, मसल पेन आपको आ सकता है, चिल्स आ सकते हैं आपकी बॉडी में. सो देयर आर वेरियस साइड इफेक्ट्स ऑफ न्यूमोकोकल कॉन्जुगेट वैक्सीन. सो इट्स नॉट दैट दे कम अप with no allergy. They do have side effects, making the third option an incorrect option. That is why we are left with option number 1 and 2. That is B is the right answer for this question. Moving ahead with the next question, question number 17. With reference to carbon nanotubes, consider the following statement. Carbon nanotubes के बारे में पूछा गया है. Carbon is an important element. कार्बन, मेक श्योर कि आप कार्बन को अच्छे से पढ़ के जाएं. कार्बन का जो भी है आप थोड़ा विकीपीडिया पेज कर जाएं, ठीक है. और आप एनसीईआरटी में से भी, एनसीईआरटीज में से भी, यू मेक श्योर दैट यू प्रिपेयर दिस एलिमेंट. देयर आर सर्टेन एलिमेंट्स यू मस्ट प्रिपेयर लाइक कार्बन, हाइड्रोजन. ये आप प्रिपेयर करके जाएं. यू नेवर नो, दे कैन आस्क यू क्वेश्चंस. सो कार्बन नैनोट्यूब, अगेन बिफोर रीडिंग or before jumping into the options, we must know carbon nanotubes क्या होते हैं. So this entire story of the carbon nanotubes, it started in 1991. There was a Japanese scientist who came up with a research paper on carbon nanotube only, and वहीं से this entire story of carbon nanotube started. ठीक है. And this carbon nanotube basically belongs to the fullerene family, and it is also an allotrope of carbon, as the name indicates. Allotrope क्या होते हैं? Allotrope basically are structurally different forms of the same element. And carbon is capable of forming many allotropes due to its valency. Like for example, it has, let me show you this, it has diamond as one of its allotrope, it has graphite as one of its allotropes, so it has many allotropes. ठीक है. All of this is mentioned in your NCERTs. Again, NCERTs are the books, NCERTs are the gem, NCERTs are the everything. Make sure you prepare them by heart, you do them by heart. So yeah, these are the allotrope of carbons, right? And if we talk about the structure of it, the structure of carbon nanotubes, ये cylindrical shape के होते हैं, consist of large molecules which are hexagonally arranged होते हैं, ठीक है. And they have a lot of applications, like varied applications, starting from the textile to the electrical appliances. Even in the medicines, they are used a lot these days. Okay? So yes, option number one is in the same lines, that they can be used as a carriers of drugs and antigen in the human body. हाँ जी बिल्कुल, we have just read कि medicine में इनका काफ़ी ज़्यादा यूज़ हो रहा है आजकल. They can also be made into artificial blood capillaries for an injured part of human body. Exactly. They can be used in biochemical sensors. बिल्कुल सही बात है. And the last one is carbon nanotubes are biodegradable. Okay. Carbon nanotubes are biodegradable. Okay. दिस कुड बी वन ऑफ दी ऑप्शन जिसका शायद आपको आंसर नहीं पता होगा और आप शायद इस ऑप्शन की वजह से कंफ्यूज हो जाएंगे. सो मेक श्योर कि कोई भी आपको एक फ्यूचरिस्टिक सी चीज़ जहाँ पर आपको दिखती है और एक पॉजिटिव ऑप्शन दिखता है, आई एम नॉट सेइंग दैट दिस ट्रिक ऑलवेज वर्क्स, बट इफ़ इट इज़ अ फ्यूचरिस्टिक टेक्नोलॉजी और अ फ्यूचरिस्टिक थिंग, सो उसके ज़्यादातर पॉजिटिव्स जो हैं सही होने के चांसेस होते हैं. सो इफ एनी स्टेटमेंट इज गिवन टू यू विच इज टॉकिंग पॉजिटिव about a particular technology or a particular thing, so there are high chances that that positive thing would be correct. So this is also correct. Carbon nanotubes, yes, if we put these carbon nanotubes through the enzymatic oxidation, they will be degraded. So yes, they are the biodegradable tubes or the biodegradable elements, right? Therefore, D is the right answer since all the options are correctly mentioned, right? These are some of the allotropes of carbon. Make sure that you prepare the carbon really well. Sometimes Wikipedia page really helps you to prepare such kind of topics, like for example carbon, hydrogen. Make sure that you prepare at least first or the second para of that particular page. कई बार directly UPSC asks question from Wikipedia page only, if you have ever noticed this. Moving on to question number 18, consider the following statements. First statement says genetic changes can be introduced in the cell that produce eggs or sperms of the prospective parent. Second statement says a person's genome can be edited before birth at the early embryonic stage. And the third statement says human-induced pluripotent stem cells can be injected into the embryo of a pig. Okay, so again, there are so many things to be understood in this question. First of all, let's start with the first the first option only, the first statement only. First statement is saying genetic changes can be introduced into the cell that produce eggs or sperms of the prospective parent. Okay, yes, it can be done. और हम इसको कर कैसे सकते हैं? It can be done using CRISPR technology. CRISPR technology के बारे में UPSC has already asked questions. So CRISPR technology is nothing but just a tool for the gene editing. So basically we can alter the DNA sequence and modify the gene function using the CRISPR technology. So this is the correct statement. Second statement is saying a person's genome can be edited before birth at the early embryonic stage. Let's see if we can do this or not. So we can do this also, and for the first time the scientists in US have repaired a disease which was causing mutation in the DNA of the early stage human embryos, and they are basically trying to create a child जिसका DNA has been tailored using the gene editing. And not just US, even the China or the UK for that matter has also tried this. Okay? Therefore this is also the correct statement. The third statement the third statement says human-induced pluripotent stem cells can be injected into the embryo of a pig. Okay, for that we must understand what are pluripotent stem cells, and before pluripotent we must understand stem cells क्या होते हैं. Stem cells again a very very important topic for our UPSC. Stem cells को आप अच्छे से करके जाएं. There are high chances that UPSC can ask you any question regarding stem cells, और इसको थोड़ा आप डिटेल में पढ़ के जाएं, because if they are asking about human-induced pluripotent stem cell, they can ask you any question which is a detailed thing of stem cells only. So stem cells, I'll tell you in simpler words, stem cells क्या होते हैं. They are the cells in the body जिनका एक specific purpose होता है. But see, we know we have a lot of cells in our body, right? हर एक cell का एक पर्पस होता है, एक स्पेसिफिक फंक्शन होता है. बट स्टेम सेल्स आर व्हाट काइंड ऑफ सेल्स? स्टेम सेल्स आर द सेल्स दैट डू नॉट yet have a specific role, and they become almost any cell that is required. And this is the beauty of stem cell, that they can become any cell which is required. ठीक है, ये कुछ अनडिफ्रेंशिएटेड सेल्स होते हैं दैट कैन टर्न इनटू द स्पेसिफिक सेल्स जिस तरीके से हमारी बॉडी को ज़रूरत होती है, जैसे जैसे हमें जो भी डिजीजेज होती है और वॉट एवर एलिमेंट्स वी हैव एंड वॉट एवर काइंड ऑफ सेल्स वी रिक्वायर to cure that disease, stem cell could be the remedy. We can produce any kind of cell using the stem cell technology, this is not a technology, but definitely there are further many technologies which are used, which take stem cells as as a thing, and modify stem cells or make stem cells which could be used in our body. Okay. नाउ टाइप्स क्या क्या हो सकते हैं? स्टेम सेल्स के 3 मेजर टाइप्स होते हैं. द फर्स्ट इज द अडल्ट स्टेम सेल्स, एज द नेम इंडिकेट्स, अडल्ट स्टेम सेल्स. एम्ब्रियोनिक सेल्स होता है. थर्ड इज द इंड्यूस्ड प्लूरिपोटेंट सेल्स. नाउ इंड्यूस्ड प्लूरिपोटेंट सेल्स क्या होते हैं? इंड्यूस्ड, एज द term indicates, induced means which जो naturally नहीं है. We are inducing something which is not occurring naturally, we have to induce it, right? So induce means scientists are creating them in the labs, and pluripotent means pluripotent means they have the ability to undergo self renewal and to give rise to all the cells or the tissues of the bodies. Let me show you this diagram. These are the classes of the stem cells, and these are the cell types. So the classes of the stem cells are totipotent cells होते हैं, pluripotent cells होते हैं, multipotent cells होते हैं. Totipotent जो है, these are the type of embryonic stem cells only. Pluripotent also comes under the same category, and they comes under the adult stem cells. इनका basic function क्या होता है? That they can differentiate into any cell type. Even the pluripotent also can differentiate into the any cell form, but multipotent differentiate into the limited range of cell type only. They cannot differentiate into any cell which is required to cure any disease. वो सिर्फ limited range तक हो सकते हैं, right? That is, it is multi, not pluri. Pluri means a wide range. Multi means a limited range, right? And here all the statements are correct, therefore D is the right answer for this question. I hope you are clear with what is stem cells, what are stem cells. I hope you are also clear that you have to go and you have to read the topic stem cell today itself, because this is a really, really important topic for the upcoming examination. Moving ahead with the next question, question number 19. With reference to the present state of development, artificial intelligence can effectively do which of the following? Artificial intelligence, again, a very important topic, artificial intelligence and machine learning. Artificial intelligence, again, as I've told you, a futuristic technology. So, until and unless a very drastic or a very extreme option is given, which is not possible, only you can put a cross mark there. Else, देर आर हाई चांसेस कि ज़्यादातर जो भी ऑप्शंस हैं वो सही होने के, जो भी पॉजिटिव्स हैं इस किसी भी पर्टिकुलर टेक्नोलॉजी के, देर आर हाई चांसेस कि जितने भी पॉजिटिव्स आर मेंशन इन योर ऑप्शंस दे विल बी करेक्ट ओनली, अनलेस एनी एक्सट्रीम थिंग इज गिवन. तो वो तो पॉसिबल ही नहीं है, बट if there are decent things are given, they will be correct only. The first thing is bring down electricity consumption in industrial unit. Exactly, it can create meaningful short stories and songs. हाँ जी बिल्कुल. it can diagnose any disease. Yes, exactly. Medical science में it is being used at a very large scale. Text to speech conversion, definitely it can do this wireless transmission of electrical energy. All of this is possible, yes. So D is the right answer for this. But let me tell you a bit of things about artificial technology. Artificial technology, in a very, very simple word, in a very, very simple language, artificial technology एक computer science का branch है, and it is concerned with making the computers behave like human. ठीक है? This is a very straightforward and a very easy definition of artificial intelligence, that we are trying to make computers behave like humans. And in other words, it is the ability of the machines to perform the cognitive tasks like human, like for example thinking, for example learning, problem solving, decision making, etc. And like I've told you, in such type of question जहाँ पर futuristic technology is present, there are high chances that mostly all of the options would be correct. Alright, so D is the right answer for this particular question. Moving ahead with the next question, question number 20. With reference to the visible light communication technology, which of the following statement is correct? VLC. Okay, this question has been taken from the Wikipedia. Agar aapne Wikipedia ka, like I have been saying it repeatedly, that you must prepare the first, at least the first para of Wikipedia for some of the topics. Agar aapne ye kiya hota, you would have marked it correct. This has been directly asked from that there only, the first para of Wikipedia. VLC, visible light communication technology. So first, before going into the options of it or the statements given below, we must understand this technology itself. So for such technology which are in news, I would want all of you to just, first of all, make your notes. Wikipedia pe jaake, just make your notes of the first paragraph, write down the important points. And second, to understand this, the visible light communication technology, ye ek wireless method hai that uses the light emitted by LEDs, okay, to deliver what? To deliver the high speed communication, which is similar to Wi-Fi only, but it uses light. That is why we call it Li-Fi. Okay, so now see the statements. The first statement says VLC uses electromagnetic spectrum wavelength jiski 375 to 780 nm hoti hai. It is correctly mentioned. This is precisely the visible light or the spectrum which the visible light uses. Then the second statement says it is known as long range optical wireless communication. This is an incorrect statement because इसका सबसे बड़ा disadvantage ही यही है. अगर आपको थोड़ा भी इसके बारे में knowledge होगा, this is the biggest disadvantage of VLC, that it uses short range optical wireless communication, not the long range ones, hence making the option or the statement 2 incorrect here. Third statement is VLC can transmit large amount of data transfer than Bluetooth. Yes, it can do. The third, it says the VLC has no electromagnetic interference. Hanji, VLC operates at optical frequencies and emit no electromagnetic interference. That is why this option is also correct. Therefore, if we eliminate 2, okay, from here we eliminate 2 from here. We are left, we eliminate 2 from here. We will be left with option number C, which should be the right answer. So, बहुत बारी तो elimination से ही we can reach to our answer. Okay? So, C is the right answer for this. All right. Moving ahead with our next question. Next question says, India में the term PKI or Public Key Infrastructure is used in the context of what? All right. So, PKI होता क्या है? What is Public Key Infrastructure? हम सबको पता है, ये basically एक कुछ सॉफ्टवेयर होता है और you can say it is a combination of software या combination of encryption technology, and that basically enables the entities or the users to protect the security of their communication, and जो भी business transactions वो करते हैं उसको कहीं ना कहीं security provide करने के लिए it works. Right? So to protect the security, to provide its entities to protect the security of the communication and business transactions network pe, right? See, you must understand that that our information is as essential as our business assets these days. Information is crucial, very highly crucial these days. So we must have some technology which can add extra layer to our security. Okay? To this most valuable asset which we are calling as information in these times. And PKI, which is Public Key Infrastructure, exactly provides that extra layer of security. So with PKI, with Public Key Infrastructure, what we can do, we can employ the advanced authentication and encryption methods to mitigate the risk of our networks, right? So what is it related with? It is related with, now I think you can easily guess, it is related with the digital security infrastructure, making A the right answer of this. Again, to deal with such kind of questions, agar aapne suna hai, only then you can deal with such kind of questions, else it is little difficult for you to reach the right answer, right? Okay, moving ahead with the next question, question number 22. Consider the following activities: spraying pesticides on crop fields, inspecting the craters of active volcanoes, collecting breath samples from sprouting whales for DNA analysis. At the present level of technology, which of the above activities can be successfully carried out by using drones. इन सब में से हम क्या कर सकते हैं using drones? Okay, so pretty much we can do all of the three which are mentioned here. We can spray the pesticides. ठीक है? There is a very comprehensive lecture which is present at the YouTube channel of Guidance IAS. I would want all of you to kindly go there and check that lecture and explore that lecture for detailed information, for detailed knowledge about drones and its various applications in the field as well as in the agricultural fields as well as the other fields. ठीक है, तो drones पे एक बहुत अच्छा lecture है, you must go and visit it. So all the three things or all the three applications are all the three things could be done using drones. Therefore, D is the right answer for this question. All right, moving ahead with the next question. The experiment will employ a trio of spacecraft flying in information in the shape of equilateral triangle that has sides 1 million km long with laser shining between the craft. The experiment in question refers to. Okay, a very dicey question. अगर आप इनमें से सब चीज़ों के बारे में आपने पढ़ा होगा और वो भी थोड़ा डिटेल वर्जन में, because this is not a direct question. As you can see, they are not asking whether this particular spacecraft or this particular mission is related with moon or related with any kind of planet. No, they are asking you some detailed thing of it about the mission. I repeat, this experiment will employ a trio of, this trio word is very important here, trio of spacecraft flying in information in the shape of equilateral triangle. Again, a very important topic, again a very important word. Then what is it related with? Let me directly tell you the answer, and then we will understand how. It is related with ELISA, evolved LISA. So, but how? For that you must see here, you must look at this diagram. Okay. ELISA, or we also call it as laser interferometer space antenna. ये एक mission है which aims at exploring the gravitational universe from space for the first time, right? What exactly is happening here? We need to understand it, and that's precisely our question is also asking. ELISA mission में or ELISA mission में, whatever you call it, it consists of a mother. This is the mother, and these are the two daughter spacecrafts. ठीक है? Spacecraft number 1 and 2. These are the daughter spacecrafts. Okay? And this is the mother spacecraft, this bigger one. Okay? So it consists of a mother and the two daughter spacecrafts. Okay? And these will orbit the sun in a triangular configuration. As you can see, it makes a triangular configuration, as you can see in this figure. So these three satellites will form a precision interferometer with the two daughter spacecraft connected with the mother spacecraft, and this interferometer will be capable of detecting the gravitational wave. This is the interferometers which are connected here, and they will be doing what? They will be detecting the gravitational wave. So as our question is asking, that it is making a triangular configuration with two daughters and one mother, and they will be detecting the gravitational waves, right? Okay, so this is the correct answer. But if we talk about these other missions, like for example Voyager 2, New Horizon, LISA Pathfinder. So Voyager 2 is aimed to probe Jupiter and Saturn and Uranus, Neptune. इनके बारे में it is related to. ठीक है? तो but on the other hand, Horizon mission is related to, अगर आपने सुना होगा, Pluto. ठीक है? Voyager 2 and New Horizon are the 1-to-1 spacecraft. तो इन दोनों options को हम eliminate कर सकते हैं. LISA Pathfinder B is also related with the gravitational waves, but ELISA is the evolved LISA, right, which fits the question the most. That is why D is the correct answer for this question. Moving ahead with the last question of the day. Question number 24. With reference to solar water pumps, consider the following statements. This is the last question for the day. Option number 1 says solar power can be used for running surface pumps and not for the submersible pumps. Okay. Second statement is saying solar power can be used for running centrifugal pumps and not the ones with pistons. Acha ji. Solar water pumps के लिए कुछ regarding questions है. See, to solve this, many candidates would have simply applied some guesswork that yes, solar energy could be used for running all type of pumps. While it may be turn out to be correct in some of the answers, but it is not necessarily that in all physics, it is not necessary that in all the physics questions and in all the electronic principles, it will turn out to be correct only. So you cannot say for sure without studying that every time our guesswork will turn out to be right. तो हमें पढ़ाई तो करनी पड़ेगी, ठीक है, बेसिक्स तो क्लियर करने पड़ेंगे. बट इफ वी टॉक अबाउट ऑप्शन नंबर 1, इट सेज solar power can be used for running surface pumps and not for the submersible pump. It is an incorrect statement. So what happens? Some solar submersible pump use a centrifugal mechanism as a surface pump, while others use a positive displacement mechanism. So submersible के लिए भी it can be used, therefore making the first statement incorrect. Second statement says solar power can be used for running centrifugal pumps and not the ones with piston. It is again an incorrect statement because even the solar power piston pump system can pump the water from a well and it delivers it to the elevated storage tank, making the second statement also an incorrect statement. Therefore D is the right answer for this, क्योंकि दोनों ही यहाँ पर incorrect statement दिए हुए हैं, right? D is the incorrect answer here. Okay, so this is the end of 2021 science and tech paper discussion, and we will come up with previous 10-year question papers year wise. And do not worry, you will sail through this examination. You just need a judicious mix of efforts and guidance. So, but efforts will be your domain and you can leave the guidance part to us. We at Guidance IAS will ensure that your efforts get the requisite direction and you end your journey with a grand success. Thank you so much.